Okay, can you hear me? Sorry, I was late. Okay, right? Can you hear me? Good evening, Habib. You can hear, right? Yes. Joel, you was absent morning, no? Uh, okay, let's start. Okay, in the morning class, we discussed the microbial productions. Microbial production. Okay, Joel, please go to recording. We discussed very important part regarding microorganisms and their characteristics and their classification, and finally we discussed microbial production part of microbial production assay. Okay, what we discussed in the morning class, we discussed microbial characteristics and their classification and their environments, and I told microorganisms living in all ecosystems. Sometimes normal organisms cannot live extreme environment, also microorganisms are living. uh, as example, hot water spring and very acidic condition, and glacier regions, inside ice as well, inside ice, inside glaciers, in polar regions, very cold temperature. Yes. Okay, now this is our next. In the second part we discussed about microbial production. In the production of microorganism, we discussed about in agriculture how use microorganism to keep the soil fertility, keep soil fertility, to keep the nutrition cycles balance, keep balance, right? We discussed about nitrogen convert into nitrate and nitrate convert into nitrogen. That's nitrification and denitrification balanced by the microorganism. Nitrification and denitrification balancing by microorganism, right? I think you can remember I told different types of nitrogen fixation bacteria. Sometimes symbiotic fixation, it is rhizobium. Sometimes freely living and nitrogen fixing. Who are the examples? Clostridium, freely living, azotobacter, and cyanobacteria. Symbiotic examples, symbiotic, rhizobium and anabina, right? Finally, I told this produce ammonium ions convert into nitrite by nitrosomonas. Finally, nitrite convert into nitrate by nitrobacter. Okay, this is a story. This is a nitrogen cycle, to keep balance of nitrogen cycle. Lot of microorganisms involved, right? What else? In production process, we discussed about bread production. In the next part, we discussed about alcohol beverage production. We already discussed 2D production, raw කොහොමද හදන්න, and beer production already discussed, wine production already discussed, and finally we discussed about vinegar production. Okay, do you have any doubt up to now? Any doubt? We discussed. Are you all collective now? Ne? හැමෝම ටියුට්ස් හම්බුණාද? සනිජ, did you collect ටියුට්ස්? Jenul, now you got? I think now you collect. Oh, Sanija, collect, right? Now, Joel, collect. Sarmia, did you collect ටියුට්? Sharon, collect, no? Good. Collected. Sonika. තව කවුද නැත්තේ එතකොට? සර්මියා? උදන්‍ය? හම්බුණාද? Sarmian Udanya. How about update? Okay, Udanya. Sarmia? Okay. Hari. Okay, let's I will revise again vinegar production, because vinegar production very important. Vinegar production. In vinegar production, it's very important. Vinegar production. I missed one point in the morning class. Vinegar production. Okay, how to produce vinegar? We have two main steps. Actually, vinegar production, we have two main steps. The first thing, any sucrose or glucose media, any sucrose C6H12O6, any glucose media, first convert into ethanol C2H5OH, C2H5OH plus CO2. That's a using Saccharomyces cerevisiae, Saccharomyces cerevisiae, Saccharomyces cerevisiae. Saccharomyces cerevisiae, right? Saccharomyces cerevisiae, that means yeast, any glucose solution convert into ethanol and carbon dioxide. Then carbon dioxide releasing ethanol dissolved in the water in this glucose solution, right? Then this ethanol mixed diluted solution is the 2D, that the product we called as 2D, at the making had in 2D, right? Raw. This 2D C2H5OH mixture, C2H5OH, 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 if it is oxidized with oxygen with Acetobacter bacteria. Who are the Acetobacter bacteria involved for acetic acid production, vinegar production? Remember the name, Acetobacter acetic, Acetobacter acetic, Acetobacter acetic, and acetic, Acetobacter acetic, and gluconobacter, gluconobacter, Acetobacter acetic and gluconobacter. What they doing? Acetobacter acetic and gluconobacter, what they doing? This ethanol convert into CH3COOH, CH3COOH, CH3COOH plus water. This water soluble acetic acid, that CH3COOH means acetic acid. This acetic acid, diluted acetic acid solution we called as a vinegar. Diluted acetic, that means acetic acid dissolved in water. This mixture we called as vinegar. Therefore, vinegar production mainly two steps. First, glucose solution convert into ethanol by the fermentation of yeast, and this ferment ethanol convert oxidized by Acetobacter bacteria, Acetobacter acetic and gluconobacter bacteria, and convert into acetic acid and water mixture. That means diluted acetic acid solution. This solution we called as This solution we called as a vinegar. This solution we called as a vinegar. Right? Normally, I told this 2D you can produce in your home. කොහොමද හදන්නේ? 2D හදන විදිය කියලා දුන්නා. කොහොමද හදන්නේ? Get a glass of water. Water glass එකක් ගන්න. Then add the 3 to 4 sugar spoons. සීනි හැඳි 4-5ක් දාගන්න and dissolve well and keep in a side 3 to 4 days. It is better to if you can add a very small amount of yeast පොඩ්ඩක් දාන්න පුළුවන් නම් the fermentation process will be rapidly. It will be rapidly fermented. If not, environment available microorganisms are growing because it's a nutrition media, sucrose media, and it's a substrate for microorganisms. Therefore, environment, atmosphere microorganisms directly growing in this solution, and they are rapidly growing this sucrose solution. Sucrose first break down by invertase enzyme and glucose. This glucose convert into ethanol and carbon dioxide within few days. Right? එතකොට 2D හදාගන්න පුළුවන් ඔයාලට. Within 3 to 4 days you can make 2D. Now in home how you can introduce after preparation of 2D, how you can introduce acetobacteria? Any method? How we can in your home? Then මේ කරලා බලන්න. Very simple practical. You only need 4 spoons of sugar and water glass. And how to introduce acetobacter acidic and gluconobacter? You know how we can introduce. අපි කොහොමද ගෙදරදී ඒවා bacteria හදාගන්නේ? How we can find this bacteria culture? Any idea? So then, how we can find this bacteria culture in our home? Do you need to buy? Joel, how we can find this bacterial culture? කොහෙන්ද අපිට හොයාගන්න පුළුවන් මේ bacteria? අපි දෙන්න ඕනනේ මේකට. Acetobacter acidic, very difficult to find environment. We have to introduce that. We have to introduce that. Acetobacter acidic, we have to introduce. Amina, how we can find Acetobacter acidic? Yeah. Any idea? Kabir, how we can find this bacteria? No, Acetobacter, not living. Azotobacter. Joel. Soil living Azotobacter, not Acetobacter. Azotobacter. Yeah, that we discussed in the morning. Acetobacter. Please go through this video, right? It is compulsory for all. Morning video, I think in the exam more than 5 marks we discussed. Definitely you can get more than 5 marks in morning video. Right? Morning video එක අනිවාර්යෙන් බලන්න හැමෝම. The soil bacteria is Acetobacter, Joel, not Acetobacter. Acetobacter means vinegar producing bacteria. Anyone? Fatima, how we can find this bacteria in your home? Very simple, me. in your home, you can get a bottle of vinegar. නැත්නම් ඔයාලා ගෙදර ගෙනාපු vinegar bottle එකක් තියෙනවා නම් it's contain huge number of bacteria, Acetobacter bacteria. You can get a small spoon of already prepared vinegar and introduce a spoon, small 1 millimeter දැම්මත් ඇති. Add only 1 milliliter of already prepared vinegar. Then it's contain enough number of gluconobacter and Acetobacter acidic bacteria. When they introduce into ethanol media, it is a very good source, good substrate for these microorganisms. Then they are rapidly growing and doing their job in aerobic conditions, and maintain aerobic condition is very important, right? Understood? Understood? Then you can perform this practical. Make Karanpula, you can conduct this practical and send me photos. Your vinegar. Can you do it, Pulanda? I'll explain again if you want. Get a glass of water glass, and add 4 spoons of sugar, and stir well, and add 2 to 3 grams of yeast. Yeast means Saccharomyces cerevisiae. It's a fungi type. Yeast, baking yeast. Yeast dala keep 3 to 4 days. Then you can see the color and smell. Using the smell and color, you can identify it's 2D now. Right? After 4 days, it's completely converted into 2D after fermentation process. Then you can introduce already prepared 1 spoon of vinegar and keep open. For what? For supply oxygen. Keep open. If you close, the Acetobacter acid will be destroyed because they are toxic. Acetobacter is completely aerobic microorganism we discussed in the morning. Therefore, they are not anaerobic conditions toxic to this bacteria, right? Therefore, we have to keep open. Then this Acetobacter bacteria doing their job and convert this ethanol into acetic acid and water solution that we called as vinegar. Understood? Understood? Kabir, clear, right? Send update. Joel, understood? Amina, Janul, Sharon? That's a vinegar production is done. Vinegar production is completed. Hari. Let's move into next production. Next production, dairy productions. Dairy productions. Okay, you know, fresh milk. Fresh milk is a good nutrition media. Good nutrition media. It's contain lactose, sugar. It's contain carbohydrates. Fresh milk. Milk is a good nutritional media. It's mainly contain water the water dissolve carbohydrates. The main carbohydrate type, remember, lactose, right? Lactose, it is disaccharide. Lactose is the main carbohydrate. Do you know? And it's contain protein as well. What is the main protein types in milk? Protein. What's the main protein types? What's the main protein type? It is casein protein. Casein protein. Casein protein. Not only that, it contains some very small amount of whey protein as well. Whey protein and casein are the main protein, right? Not only that, it contains vitamin, essential amino acid, vitamin, some minerals, right? Some minerals. The milk is a good nutrition media. It's a very good nutrition media, right? Sorry. Okay. Dairy production. Milk is a very nutritious food. It is nutrient medium containing minerals, proteins such as casein, fats. I forgot fats also there, and sugar such as lactose. That means main carbohydrate type is lactose. Main carbohydrate type is lactose. Milk, right? Milk. And vitamins contained in aqueous medium. That means aqueous medium mean water soluble. All compound dissolved in water solution, right? And the milk is, remember, inside the body of milk, inside the body, that means inside the mammary gland, this milk produced by mammary gland, right? Inside the body of cow, there is no any microbial attraction. It is sterile, not sterile, but we called no any microbial infection, right? It's completely, I forgot this word. Sterile. Sterile. It's completely sterile. No any microorganism. No any microorganism. But due to these highly nutrition characteristics, highly nutrition property, when it is released from the body, when it is released from the mammary gland, huge microorganisms quantity in the atmosphere get that subtract as get deposit and subtract as the milk, right? The milk produced by the mammary gland, it is sterile, but secreted out is inoculated with microorganism as shown due to its nutritional property. Due to this nutritional property, this deposit and involving microorganism, what they doing different breakdowning process. They are protein breakdown, carbohydrate, lactose breakdown, and due to that, it will be affect to the lifetime of the milk. That why in normally the lifetime of fresh milk, if it is open environment, open atmosphere, it is only 1 day. Next day we can't use. That's why lot of microorganisms rapidly growing on this highly nutrition media, right? That's why the lifetime of fresh milk, it is only 1 day. 1 day. Within 1 day, it will be fermentation, right? Lot of changes. It's a breakdowning their compounds and lot of changes, right? By carefully controlling this naturally occurring process, by carefully controlling this naturally occurring microbial attractions, dairy products such as curd, yogurt, cheese, we can produce by controlling these natural decompositions, natural microbial attraction. Okay, curd production, very simple. Curd production, very simple process. Curd production, curd production, curd. Curd production. Curd production. Okay. How to produce curd? Curd producing by fresh milk, no? Now, as I know earlier, the curd fresh milk is a good nutritional media for all microorganism type. It's a good substrate. It's a good substrate. Therefore, when it is released from the mammary gland, when it is released from the cow's body, animal body, it's a lot of microbial attraction, fungi type, protozoa type, lot of bacterial types growing on this substrate due to its nutritional property, right? Now, if we produce curd by these vulnerable environment available microorganisms, it will be different taste and lot of byproducts. Some proteins can be breakdown and lactose can be breakdown due to their characteristic. Therefore, we need to introduce specific microorganism for curd production that we call as lactobacillus microorganism. Lactobacillus microorganism need to be introduced to production of curd, right? It's very simple. The first thing, fresh milk pasteurization. Fresh milk pasteurization mean just heating. Normally we heat up to 90 Celsius and keep 20 minutes. 20 minutes for pasteurization, naturally available microorganisms destroy. Right, 99% we have to destroy. We can't 100% destroy because some thermopilus bacteria retain in 100 Celsius as well, no problem. 99% of bacteria we can destroy by this pasteurization process. Pasteurization means heat and destroy naturally growing microorganisms, right? Naturally growing microorganisms we can destroy. After that, we have to introduce when it is cool down. After pasteurization, we have to cool into room temperature. And need to be introduced. Introduce karandone lactobacillus special culture, lactobacillus bacteria. Okay, how to introduce this lactobacillus bacteria? Normally, what we doing for introduce lactobacillus bacteria? It is same as vinegar production. What we doing? What we doing? How we introduce lactobacillus bacteria? Yes, adding prepared spoon of normally 50 milliliter of already produced curd sample. Then it's contain enough lactobacillus bacteria. Then we can mix this solution and keep for incubation. Incubation means we have to provide uh, sufficient required conditions for the for this microbial growing, right? Temperature, uh, normally the temperature and moisture conditions, relative humidity, and the pH values, everything keep given according to the requirement. Given according to requirement. Then incubation. In the incubation process, incubation means provide incubation. Provide incubation. Incubation means provide favorable condition for microbial growth. Provide favorable condition for microbial growth. Okay, now how to produce curd? Get fresh milk. It's contain huge amount of vulnerable microorganisms, naturally environment microorganisms. Natural microorganism came from environment, and the first thing we have to heat milk and pasteurization. After pasteurization, we can get into room temperature again. Why we need to get into room temperature? Without get into room temperature, in high temperature, if we introduce lactobacillus, they will be destroyed, right? They are not tolerant to high temperature, lactobacillus. That's why we need to cool up to room temperature, cool to room temperature, room temperature, and introduce lactobacillus culture. What we doing? Already produce sample of milk, curd, not milk, curd sample එකක් තමයි දාන්න. Then incubation means provide favorable conditions for lactobacillus growth. Okay, in the incubation process, what happened? It is very important. The lactose sugar, the lactose is the main carbohydrate, right? Lactose sugar contained in microorganism, sorry, not micro, lactose sugar contained in milk, fresh milk, convert into lactic acid. By lactobacillus microorganism. Okay, lactose sugar convert into lactic acid by lactobacillus microorganism. In the lactic acid production process, what happened to the pH value of the milk? What happened to the pH value of the milk? pH value getting low. pH value getting very low. As a result of pH value getting very low, what happened? It's become acidic. In acidic condition, what happened to the casein proteins? Casein proteins denaturate. Casein protein denaturate. Casein protein denaturate. That's the story. In the low pH value, casein protein denaturate. In the denaturation of proteins, what happened? Casein, liquid casein convert into crystals, solid. That we called as curd, semi-solid curd. Understood? Any doubt? That's the story of curd production. That's very important. The curd semi-solid texture is formed due to the coagulation of casein as a result of denaturation under low pH value as a result of lactic acid production. Lactic acid production. Sorry. Yeah. Okay, that's the curd production steps. First, pasteurization of fresh milk and hold down into room temperature and introduce lactobacillus bacteria, curd, pre-prepared curd sample, and uh, incubation provide favorable condition for microbial growth. In the incubation period, what happened? In the incubation period, lactose sugar convert into lactic acid by lactobacillus bacteria. Right? This lactic acid getting pH very low in the solution. As result of this low pH value, casein protein in the milk coagulate by denaturation. As a result of denaturation of casein protein, they are coagulate solid venom. As example, do you know eggs? What happened? Eggs boiling, normal albumin protein, liquid form, egg, right? But when it is heating, denaturate and convert, coagulate. Egg get coagulated on same. In the denaturation of casein, what happened? Liquid form to coagulate semi-solid form. Understood, Kabir? That's the milk production process. Milk production process. Milk production process. Understood? Sharon? Done? Okay. Let's try to complete curd production. Today, I'm very difficult to speak. Hari, ada අපි පුළුවන් තරම් ඉවර කරමු. Tomorrow morning again you have class. Tomorrow morning I hope to complete this lesson. Yeah, today little bit difficult to continue. Hari, පුළුවන් තරම් කරමු අද. Okay, the curd production is done. Okay, the next dairy production is yogurt production. Yogurt production. Yogurt production. The yogurt production also, it is same first step, get fresh milk and get fresh milk and get fresh milk and we have to pasteurize. Pasteurization mean heating, normally 90 Celsius, 20-minute heating. In the heating process, all naturally available microorganism what happened? Destroyed. Right? Naturally available microorganism will be destroyed. Okay, after pasteurization, the same process, right? Same process as curd production. Right. After pasteurization, we need to get into room temperature, same as curd production, room temperature. Otherwise, this cultured microorganism can be destroyed. We have to introduce gelatin and pre-prepared yogurt sample. Pre-prepared yogurt sample mean it contains lactobacillus bulgaricus. It's a special microorganism, lactobacillus bulgaricus. Lactobacillus bulgaricus and Streptococcus thermophilus. Thermophilus. Streptococcus thermophilus, Lactobacillus bulgaricus and Streptococcus thermophilus, they are the microorganism involved for fresh milk, that means curd, lactose sugar convert into lactic acid, the same process, right? Same process, lactose sugar, lactose sugar, lactose sugar convert into lactic acid, lactic acid. And yogurt production, other thing, we have to introduce some part of sugar. I forgot. Sugar dana. We have to introduce sugar, gelatin, and lactobacillus culture. Lactobacillus culture means pre-prepared yogurt, not curd. Curd are not curd done bad. Why? Curd contain only lactobacillus types, but we need special culture now for yogurt production: Lactobacillus bulgaricus and Streptococcus thermophilus. This Lactobacillus bulgaricus and Streptococcus thermophilus are living in already prepared yogurt sample. Then we can get sample of spoon of yogurt and introduce bacteria and add gelatin in required amount and sugar required amount. In the finally the incubation same as previous. Incubation. In the incubation process, what happened? Lactose sugar converted to lactic acid by this bacteria, by Streptococcus thermophilus and Lactobacillus bulgaricus convert lactic acid into lactose sugar, that's carbohydrate, main carbohydrate in milk, convert into lactic acid. Then what happened? It's getting very low pH value and acidic. pH value getting very low and it is acidic. Now what happened? Caseins denaturate and liquid casein convert into coagulation. As a result of coagulation, it's converted to solid, but their texture getting gel-like structure due to this gelatin, and it is more sweet in taste than curd, because we adding additional sugar. Therefore it's sweeter than curd. Okay, that's the difference, and its texture is very soft due to adding gelatin. Gelatin turns as softica, like gel, right? Okay, that's the yogurt production. Any doubt? That's the yogurt production. Any doubt? That's the yogurt production. Any doubt? That's the yogurt production. Amar, Fatima, understood? Any doubt? This, you have remember this casein denaturation process. Why casein denaturation? Due to low pH level. In biomolecule lesson, I think you can remember. I hope you can remember. There are three main factors to protein denaturation: high temperature, pH value, that means acidic and base conditions, and heavy metals. Any factor, proteins, due to this any factor, protein denaturation. As example, egg boiling. Normally egg albumin in liquid form, if it is boiled, heated, it's denaturated, albumin coagulate. ඒක තමයි egg boil are solid form. Boiled eggs are solid form. Okay, let's move into take cheese. Let's move into cheese. Cheese production. In cheese production, the main difference is we have to introduce for fresh milk. Pasteurization. The first is pasteurization, same as yogurt and curd production. heat kala pasteurize karana. After that, we have to introduce renin enzyme. That please highlight renin enzymes. Renin enzymes. We have to introduce renin enzyme. This renin enzyme, what they doing? What they do in this renin enzyme? Casein and other solution, casein protein and other solution will be separated. See, milk coagulation process use the coagulated milk. Coagulated mean casein, right? In cheese production, mainly use casein. Casein is separated from the milk. This soft, semi-soft and hard cheese are made by this separated casein. Okay, actually this cheese is separated casein. Different shapes we can see separated caseins are the cheese. First, pasteurization and renin enzyme renin, right? They are the different dairy productions. We discuss curd production, yogurt production, and cheese production. Okay, let's move into compost fertilizer production. How to involve microorganisms for compost fertilizer production. Hari. Okay, what are the main raw materials for compost fertilizer production? Yeah, dry leaves, Diricidia leaves, and cow dung, and garden waste. Yes, garden waste, kitchen waste we can use to compost production. Actually, the compost production is a decomposition of this organic material, using microbial decomposition, right? Now, after organic material decomposition, it's producing brown color crystals. They we called as humus. I think BST students already know what's the meaning of humus. Humus are the compounds that is very important to plant growth and to keep the water holding capacity of the soil, to keep the water holding capacity and the nutrition capacity of the soil, to keep the nutrition capacity of soil and water holding capacity, the water retained in the soil, because these humus retain absorb huge amount of water and nutrition, and it is involved for microbial activities development. Then the soil structure will be developed, right? There are a lot of application of this humus. To supply this humus, organic fertilizers, that means natural compost, are very important to increase the humus level of the soil. Then functions of humus: reservoir of mineral salts, and it is retain water, help maintain the pH value of the soil. It's improve structure of soil by binding together with soil, forming granules, and improve soil aeration due to the structural development, and prevent oxidation and sodium salt. Remember, favorable conditions for compost production. Normally, in normal condition, natural conditions, if you decompose organic material, මේ වගේ අපි හිතමු garden waste, think we have garden waste lot, we need to decompose that without any control condition. If we're doing in normal natural condition, it take normally more than 2 months to decomposition. But remember, if we maintain control conditions well, we can produce compost within 3 weeks, 21 days. Okay, they are the favorable conditions for compost production. They are the favorable conditions. The first one, the temperature. Temperature. Sorry, first one, carbon nitrogen ratio. In the raw material, carbon nitrogen ratio need to be maintained 30 to 1 minimum. 30 carbon to 1 nitrogen. What's the meaning of 30 carbon to 1 nitrogen? Mean, dry leaves, straw, they contain mainly carbon. BTCDR leaves, cow dung, the leguminous plants, soybean type leguminous plants, they provide nitrogen mainly, right? In the raw material, we have to get in this ratio. Again, 30 kilogram of straw danana, 1 kilogram of cow dung, like this. Understood? We have to maintain the carbon nitrogen ratio of the mixture 30 to 1. 30 to 1, right? The second characteristic, temperature. Temperature. Temperature 43 to 65 Celsius. That means more than room temperature. Samani 50 wage perfect for this microbial activation. Right? Temperature 43 to 65 Celsius. Remember, moisture level, that means water, moisture level 40 to 45, 65%. Again, 50% forget idea, right? 50% of moisture level is ideal. And pH value neutral wage tiyenna neutral. Close to neutral, 6.5, 7.5, right? Okay, they are the main requirement, favorable conditions for microbial activity. Hari. How we can provide these requirements? We can cover. Sorry, pH it's not a main factor. Write down oxygen, 5%. 5% of oxygen. pH is not the main value. pH important mainly for sorry. Okay, I told you they are the main favorable conditions. What are the conditions? Carbon nitrogen ratio 30 to 1, temperature 33 to 45 Celsius, and moisture levels 40 to 65 Celsius, and 5% of oxygen minimum. Not minimum 5%, at least 5% oxygen is should there. Okay, now think this is a organic material pile in natural condition. Maybe both it's take more than 2 to 3 months to decompose it, to produce humus. But if we can give these favorable conditions, within 21 days, 28 days, we can get compost. We can accelerate this process, decomposition process. Okay, what we can do? This is an organic material pile. Okay, we can close this by අපිට මේක close කරන්න පුළුවන් using black polythene. Black polythene. First thing, we have to maintain the carbon nitrogen ratio 30:1. Okay, so I told you they are the main favorable conditions. The first thing, we have to select raw materials in carbon nitrogen ratio 30:1 ratio, right? That means 30 kilogram of straw and dry leaves, 1 kilogram minimum nitrogen material, cow dung or glycidia leaves, like this, right? Then the ratio is done. Ratio is done. Ratio is done now. Then we can cover this mixture by using black polythene and completely seal now. Completely seal kala thiye, right? Completely seal. Then we can maintain inside temperature, right? Inside temperature, and we can before cover we have to introduce 50% of water, 50% of moisture. Moisture api dala thiye. And how to introduce? Now it is completely closed. Then how to introduce oxygen 5%? Initially, it contains some percentage of oxygen, no problem. After 7 days, once a week, you have to rotate this mixture, open and rotate, and again close to introduce oxygen and to mix these microorganisms uniformly, evenly through the mixture, right? Then we can, after 3 times separate, after separate 3 times this mixture, we can get finalized compost. Right, this is a compost production. There are the different types of methods we can use to compost. Composting beam method, pit method, and Jiva Kotu method, and rotating bin methods. Okay, this is a barrel method, barrel concrete production. That's a this is a pit method, and this is rotating drum method, and that one also pit, that we call as pile method. As a pile, we have to cover completely by black polythenes. Right. That's compost production. What are the favorable conditions? Temperature 43 to 65, moisture 40 to 65%, oxygen 5%, and carbon nitrogen ratio 30 to 1%, 30 to 1 ratio. Any doubt? Remember, let's move into use of microorganism for bioremediation. How to use microorganism for bioremediation? Okay, what's the meaning of bioremediation? The bioremediation mean is the conversion of toxic and hazardous compound in the atmosphere convert to non-hazard material, non or very low hazards compound by using microorganism or any organism, right? This is a bioremediation. Bioremediation means pollutants in the atmosphere and environment. Pollutants in the atmosphere environment converted low hazard compounds using organisms. In this process, in this part we discussing how involve microorganism for the bioremediation process. කොහොමද microorganisms ලා use කරන්නේ for bioremediation process, right? Microorganism how involved for bioremediation process. Okay, now this bioremediation mainly 2 types: natural bioremediation and artificial bioremediation. Natural, what's the meaning of bioremediation? Any pollutants or toxic compounds in the environment convert into non-hazard compounds using organisms. That we can use animals, plants sometimes use, microorganism use. In this lesson, we discussing how to use microorganism for this bioremediation process. Understood? Habib. The bioremediation mean any toxic compounds in environment convert to non-hazardous compound using organism, plant or animal or microorganism. In this part, we discussing how use microorganism for the bioremediation process. This bioremediation process mainly 2 types: natural bioremediation and artificial bioremediation. Natural bioremediation and artificial bioremediation. What's the meaning of natural bioremediation? Natural bioremediation means pollutants in the environment controlled by naturally available microorganisms. ये can naturally mean microorganisms के use करना. As example, naturally environment available microorganism using. Example, release of nutrition source containing carbon, nitrogen, and phosphorus to the relevant locations to reduce pollution seawater due to oil spills. In natural bioremediation research has shown that Pseudomonas species, see, Pseudomonas is naturally available microorganism living in soil or water are affected to decomposition of toxic chemicals, heavy metals especially. This Pseudomonas decomposition of heavy metals, that's natural. Pseudomonas in natural available microorganisms, heavy metals break down. That's a natural bioremediation. Natural bioremediation. Okay, then what's the meaning of artificial bioremediation? Compost production also natural bioremediation, right? Naturally available microorganisms used to decomposition of organic material. Now, what is the meaning of artificial bioremediation? In artificial bioremediation, cultured in laboratory cultured microorganisms, artificially cultured microorganisms, artificially modified microorganisms used for breakdown or for whatever chemical toxic compounds. The number of organisms capable of degrading toxic chemicals is very small and their growth rate is also very low. For this reason, transgenic bacteria mean using genetically modified technology that we called as gene engineering technology. Genetically modified bacteria producing with large capacity and long lifestyle. Examples. Examples are normally ships in the sea produce oil spill spots in the sea. Normal available microorganism decomposition rate is very low. Normally available microorganism, this hydrocarbon decomposition rate is very low. Therefore, using genetical modification, we can increase microorganism. We can produce microorganisms with higher decomposition rate of hydrocarbon. Then oil spills in the water rapidly break down. The pathogenic strains produced in this way have a unique ability to grow by decomposition mineral oil. In this way, using genes obtained from microbes that break down toxic chemicals. Yes. Okay. Have it. Now, wastewater treatment. Okay. Now we discussed up to bioremediation. Okay, wastewater treatment. Let's discuss tomorrow morning. Tomorrow we start 8 a.m. We will listen in Patangam. 8 a.m. I need to complete this lesson tomorrow. Now 50% is complete today. No? Now we discussed up to wastewater treatment. Yeah. After wastewater treatment, directly we can move into plant stems. Yeah, 50% complete. 50% complete. Remain parts are very simple. Alright, let's start tomorrow morning. Tomorrow, 8 a.m. Right? 8 a.m. Don't miss anyone. Next week, not next week. Tomorrow, I will discuss the plant primary growth, secondary growth, and their changes, cambium tissue changes, everything we can discuss tomorrow. Plant stem structure, root structure, leaf structure, internal structure, and their characteristic and their changes with growing. Okay, everything we can discuss. They are very important. Okay, let's meet tomorrow morning. Today may finish a little bit early because I'm very difficult today. Okay, let's discuss tomorrow morning. Goodbye.